Thank you everybody for coming, and thanks Giovanni for giving me this opportunity to show you the latest thing we have been doing at the ALBA Synchrotron in Barcelona regarding imaging of 3D nanomagnets with X-rays. So this is of course a group effort, and I have shown here the people that work at ALBA, but also many users and collaborators have been involved in these developments, and I will thank them also at the end. In the top, you can see here a drawing which basically sums up my talk, which just tells that if you have a more and more complex object, you have to see it from more and more different angles to understand really the shape that it has. And in our case, not only the shape, but also the three-dimensional the direction of the magnetization in 3 dimensions. I will do a very brief introduction of what we call 3D nanomagnetism, this emerging field that is getting quite a push in the last years. Also a very brief introduction for those of you who are really far from the field of how synchrotron X-rays can help you understand magnetic materials in general. And then I will go on to the 2 full-field X-ray imaging microscopes in operation at ALBA, but not only at ALBA, but I will present what we do at ALBA, and where one can look at small 3-dimensional magnets with soft X-rays, which is a PEM, a photoemission electron microscope, and a transmission X-ray microscope. So in general, 3D nanomaterials are playing increasingly an important role for nano devices. As compared to 2D, they are much more complex to prepare, process, and integrate, but they have the advantage of having a lot of active area, more possibilities for interconnectivity of scaffolding. Just by the shape, you can understand that. And also, in the case of nanowires and nanotubes, which are some of the objects that I will show you today, the length and the shape permits engineering, for example, the mechanical torque or adding different functionalities to different segments, changing the shape or the curvature with some external field or chemical stimulus, etc. So they have, in general, 3D nanomaterials or 3D composites, many possibilities in the future. In particular, when we talk about magnetic ones, nanomagnetism refers to when the sizes and shapes approach the tens to hundreds of nanometers lengths, which are already relevant for magnetic interactions such as exchange. So the material, so to speak, will feel the curvature, for example. This gives rise to new physics. For example, we can have curvature-induced chirality or even DMI without heavy elements. We have special types of domain walls, which are not possible in 2 dimensions, with different limits for speed, even different speeds for right or left-hand side chirality or propagation direction, and so on. Here I show a very simple example of a type of domain wall in 3 dimensions, the block point domain wall, which only exists in thick objects, let's say, while in 2 dimensions, in stripes, the typical domain walls are transverse and vortex-like. And in the end, one can see that adding the 3rd dimension, that they share the same topology, and they are the same. They coexist, let's say, in nanowires. But there is a different type where there is a block point with 0 magnetization at the center, necessary to maintain continuity of the vector field, while diminishing the electrostatic energy by keeping the magnetization parallel to the surface in the circulation. And this type of domain wall in wires that does not exist in stripes can propagate much faster because it doesn't suffer these transformations which make us reach the Walker breakdown. This is just a simple example that can help visualize how the 3rd dimension adds different topologies or functionalities. Of course, always when you have new physics, one can open new applications. This boom in 3D nanomagnetism is possible to a great extent due to the advances in new preparation methods, be it ion deposition aided by a focused electron beam to photolithography, etc. I like this example very much because it shows by a relatively simple electrodeposition route, one can make even an interconnected, so a 3D interconnected assemble of magnetic wires. New materials must be accompanied by new characterization tools, both at the computational level, so aided by high performance computing, and sometimes having to go to hybrid methods, not only micromagnetic simulations, but hybrid atomistic continuum methods. And of course, new characterization tools from a experimental point of view, of which some are based on electrons, like electron holography or Lorentz electron microscopy. Others are modifications of methods that were developed for 2 dimensions, like Moog microscopy or SQUID or MFM, and some are based on X-rays, as I will show. Why are X-rays advantageous for some problems? Of course, you don't need a synchrotron for every problem, but for some problems you do need a synchrotron. One very important aspect is that they have tunable wavelength, so one can choose at which element one resonantly probes. Not only which element, but even, for example, when one has different cations coexisting in a structure, which oxidation state or in which atomic position one is addressing, and follow the whatever hysteresis curve or magnetization reversal specifically for one kind of cation. They are extremely brilliant, so what enables looking at small quantities of sample and doing with reasonable acquisition times, or very dilute samples. They are polarized, and the polarization is variable with almost everywhere, most synchrotrons. So one can look at contrast that depends on orientation of orbitals or of magnetization, for example. This is the so-called magnetic circular and magnetic linear dichroism, where the absorption of a certain element is slightly modulated by the relative orientation of the X-ray propagation and the magnetization of that element. And synchrotrons, the X-rays are produced in short pulses. This is the time structure, for example, at ALBA is 1 to 20 picosecond pulse every 2 nanoseconds. Every synchrotron is a bit different. Also, there are sometimes special working modes with one taller pulse among the other ones, and so on. This is depends on the characteristics of each machine. But this means that almost for free, let's say at a synchrotron, one gets the time resolution of the narrowest pulses, plus some jitter. And this is profited for by doing a pump probe spectroscopic experiments. So this must be experiments where the process is repeatable. In order to have enough signal, one has to repeat many millions of times the same process. Another thing that is very interesting for nanomagnetism and X-rays is that we are talking about the length scales, tens to hundreds of nanometers, where exactly materials are partially absorbing and partially transmitting X-rays. So we will be able to probe the whole sample. Let's say it's not bulk, but the whole interior of the sample, let's say. So the 2 microscopes I will show you as examples today are both full field, meaning that we do a parallel imaging of a micron-sized area with tens of nanometers resolution. But they are very different in the sense that in the PIM, in the X-PIM, one makes the image, is an electron microscope, one makes the image with photo-emitted electrons that are emitted as a consequence of X-ray absorption. And in the transmission X-ray microscope, one does the image with X-rays, so directly, with the X-rays that are not absorbed. So the differentially transmitted X-rays, one makes the image of the sample. So they are very different methods, but they can be used to probe the same samples in many cases. I will start with the XP, which is actually, it's a surface microscopy technique. It's very surface sensitive. This is the typical microscope that is at many synchrotrons, where we can see, apart from illuminating the sample with an X-ray beam and then making an image with the electrons, one has many other intermediate and accessory elements, like an electron gun for topography imaging, several lenses to change modes from imaging to diffraction, for example, an electron energy analyzer to do images with filtered electrons by energy, and so on. So it's a typical surface science machine where one can combine different techniques, but also the typical resolution is in 30, 50 nanometers, depends a lot on the machine and how well it is aligned. It can go down to 20 nanometers, sometimes it's limited to 70, depends a lot of things. How can we look at objects that have a third dimension? We do this by so-called shadow beam. So we look at simultaneously at electrons that are photoemitted from the sample surface, in this case a wire. This was the pioneering work in 2011 done at BESI in the microscope where Florian Kronas leads the group. One looks simultaneously at photoemission from electrons from the surface of the wire and electrons that are emitted from the substrate after being excited by X-rays that have traversed the nanowire, including the core. So they carry some information about the core. This is an electron micrograph of this first wire that was shown. It is magnetite wire, partially filled with nickel. And one can see here the PIM image. This is the emission directly from the wire, and there is a shadow on the background, and one can immediately see that the shadow is much darker where the nanowire is filled. And not only that, but one can see in the emission here, one can see the iron spectrum. The transmitted intensity in this region is an inverse of the iron spectrum. So it's a transmission iron spectrum. And here we can see both a transmission iron and a transmission nickel spectrum. And of course, this spectra, the matter-light interaction is the same. So these spectra also carry dichroic contrast. So they carry information of what was the magnetization in the core of the nanowire. In some cases, with very good resolution, one can even see more parts of so even a part of transmitted intensity already within the nanowire itself, like in this very nice example with many. This is the direct image. You can see in the direct image, one only see in the, let's say in the sum, in the elemental image, one only sees, so where is the cobalt? The cobalt is here, but in the shadow on the substrate, one can see also magnetic contrast, no? And then recalculate back what the inside of the wire looks like. And it's amazing what growers can do nowadays. For example, you can see you can see that it's very different, the aspect of the contrast in the shadow when one has a curling magnetization or a transverse magnetization. So it's amazing that one can do even this kind of wires with crystalline material where the crystalline anisotropy overcomes totally the shape anisotropy of a very, very elongated wire. No? One can also fabricate modulated in diameter wires, modulated in composition, etc., to imprint certain characteristics. In this case, for example, nanowires with chemical barriers were demonstrated that the chemical barriers were pinning centers for domain walls, and also the role of chirality, knowing the importance of the role of the chirality of the relative orientation of the curling magnetization at the surface and the axial magnetization of the core, the walls where chirality is not switching, they have a displacement, a reversible displacement with small magnetic fields, always pinning at these notches, whereas the others require a much higher magnetic field to be depinned, where the chirality must switch, okay? So this is something one can only understand knowing the magnetization of surface and core of the wire. And this here to the right is an experiment of moving domain walls with current pulses instead of with magnetic field. And here is, well, it's not so hard to understand just from this, but from many statistics of many moving walls, the group around Olivier Flouchard in Spintech in Grenoble were able to demonstrate that, in fact, the Ørsted field associated with the current pulse favors that all walls transform into block point domain walls once the pulses are applied, and this makes that the motion is not limited. One moment. Is not limited in spin by the Walker breakdown as for 2D strips. So this was the first. This is only from last year, this demonstration. It had been predicted. So these are some of the unique things one can do with nanowires. Of course, another important thing of X-ray imaging is that one has relatively more space around with as compared to nanosquid or MFM to scanning probe techniques. And also, one can apply some fields at the sample position, less in PIM and more in TXM, we will see in a moment, because the electrons are affected by the fields and the photons not. But already one can have quite a few functional sample environments, varying temperature, fields, pulses, electric field strain, and so on, which is much harder in other experimental setups. So these are just some examples of possible sample environments that already exist at ALBA and that any user can access if the proposal is granted. So in summary, in photoemission, one has extreme surface sensitivity and can work with very small amount of materials, and one can combine in most instruments absorption contrast with several other surface science techniques like low energy electron microscopy or XPS. It's easy to implement variable imaging temperature and in situ sample preparation, and one can look at objects that say 100, 200 nanometer thick, thanks to this contrast that pervades on the shadow of the objects, no? And then one can look at it in the photoemission from the substrate. It has some limitations, so the samples must be rather flat and conductive, compatible with ultra-high vacuum. Between the sample and the objective, there is high voltage, not like in really high voltage machines, but 10 to 20 kilovolts. So there is always some risk of discharges with elevated objects. The applicable fields are limited by imaging artifacts, and this a full rotation of the sample in order to look from different angles at the same object and obtain the full 3D magnetization vector is slow. It's possible, but it's slow. Let's move on to the TXM, the transmission X-ray microscope. In this case, it's a full field. In many synchrotrons, there is a scanning TXM. This one is a full field microscope. The beam, the X-ray beam, is focused onto around 12 micron diameter area on a sample by a glass capillary. And then an image of the sample is done in transmission with the help of a Fresnel zone lens, so a diffractive lens. The sample is on a very high precision rotation stage. This was a microscope that was built for cryotomography of cells, so bio specimens. So it's all, let's say, set up for cryo. You can see in this picture, you can see this is the capillary. And this is the zone plate. These are retracted. In measuring position, they are much closer. The zone plate is a couple of millimeters, and this is 1.5 centimeters or so. And this is an optical microscope for correlative microscopy. The samples are typically mounted on this type of stubs, so on very small grids, the typical TEM grids. In 3D, the resolution is 50 nanometers into the 30 in the best conditions. And for magnetic contrast in this beamline is called Mistral, where there is no undulator. One can either shift a vertical slit to select the upper or lower part of the X-ray beam coming from the bending magnet, or one can look at different absorption edges, or now we are developing a bump in the storage ring around this bending magnet in order to keep the optics fixed, which will increase the quality of the beam even further. The typical substrates that one use, they can be from the same ones used for biosamples, so typical TEM grids with a thin polycarbon film, quantifoil, whatever. Very often silicon nitride membranes for flat samples for multi-layers and so on. And one can even use this kind of omniprobe holders made for 3D objects where they are totally freestanding within vacuum then. When looking at the sample, we saw at the beginning that the transmission of X-rays or the absorption of X-rays, so both at the absorption edge of a magnetic element will have a part that depends on the relative alignment of the beam and of the magnetization. So if one can rotate the sample, around one axis, one can already obtain two components of the magnetization vector throughout the whole thickness of the sample. The pioneering experiment for this, if I am not mistaken, was this work on tubes of rolled up magnetic membranes done in ALS. Peter Fisher and colleagues in the Advanced Light Source in California. And you can see very closely followed by some similar 3D magnetization determination, but in multi-layers. So these were multi-layers with different magnetic materials where thanks to the rotation to different angles, one could see where there was a block point or how the magnetization reversal happened at these branching points of the stripe-like domains. Then going on to full tomography experiments. So for a full tomographic reconstruction, one needs to rotate around two axes for the full 3D. It was first done at the Swiss light source with hard X-rays and 100 nanometer resolution, and is now done also at ALBA with around 50 nanometer resolution with soft X-rays. And this is an example of the last publication from ALBA, which is very nice. It's just to show that just the geometrical curvature and helicity can imprint chirality on the magnetization. And something that I like a lot about this publication also is that, as you can see here, are collaborating people who were pioneers in different synchrotrons of the world. Now they are working together, like Claire Donnelly, Aurelio Hierro, Amalia Fernández Pacheco, Peter Fisher, Salvador Ferrer, Eva Pereira, Andrea Sorrentino. So this is really a community that is working quite strongly together. This is something also very nice of this field. For some of these experiments, also functional sample environments are necessary, sometimes just a big pulse to invert magnetization, and sometimes electric fields and so on, that are all sample environments that are still in development at Mistral, because as I said, it was built for biology, no? But it's coming up. In summary, with TXM, one can study layers that are buried quite deep. Imaging is not perturbed by external magnetic fields. One can look at fully freestanding objects, and flatness, conductivity, or surface cleanliness are not so critical. And let's say the minimum amount of material that one has to look at is a bit larger than in PIM, because one looks at the full thickness, and one needs a transparent substrate. And at least at Mistral, the measurement temperature is fixed at either 100 or room temperature. And changing the temperature during measurement, it causes a lot of drift, so alignment is not very good, because here this depends on the mechanical stability of the microscope mechanics. The work I have shown is thanks to many people. In particular, a lot of support from electronics, controls, technicians, and mechanical engineering at ALBA. And of course, the collaborators and users who are constantly posing new challenges and new ideas. Thank you all for your attention.